Hi everybody. So I think I'm recording a video after a long time. So this video is going to be a bird's eye view of a few areas, and it is also an area that I'm currently working in. I'm employed uh, in the industry in this area, that is computational chemistry, computational biology, cheminformatics, bioinformatics, and the new emerging area of applying data science and artificial intelligence to these areas. So if you see my channel previously, it focused on a lot of physics videos. My education itself was completely in physics, BSc, MSc, MPhil, PhD. They were all in physics. So initially I wanted to spend a little bit of time of brief reflection of transitioning from physics to these new areas, and also try to gather a bigger than personal picture of this transition. So if you see, look back in history, a little bit of history, the 20th century, people say it is the century of theoretical physics, because right at the beginning, close to the end actually of the 20th century, and right at the beginning, we have the Maxwellian revolution. Then you have the electrical revolution, what brought about the electrical revolution, electricity, and then you have Einstein's revolutions, the special relativity, general relativity, and what followed was the subatomic particle, atomic and subatomic particle revolution, the quantum revolution, and so on. So the 20th century is largely gathered as the century of theoretical physics, and at this high time of theoretical physics, people were making statements like, only theoretical physics is a science, and everything else is stamp collection, because only in theoretical physics can you explain why something happens the way it is, because you have a theory to explain it. But in other disciplines, you are just basically collecting data. So that's the kind of opinions that were actually voiced out. And close to the end of the 20th century and in the beginning of the 21st century, we see that at this time period, slowly there is a stagnation of in terms of we don't have new experimental phenomena that is not explained by existing theories. Instead, what we have is more and more theories explaining the same phenomena. So at a fundamental level, we have not made great progress in theoretical physics, because at a theory level, at a fundamental theory level, we have not made great progress, because there is, at a fundamental level, we don't have new phenomena, because our experiments have not reached a stage where we have explored new phenomena which is not explained by existing theories. But instead, what has happened is theoretical physics has branched into other areas to explain in the theoretical mechanism in many areas. So here you have in chemistry, when computers started coming, people saw the potential to use quantum mechanics to solve problems in chemistry, to understand at an atomic level what the reactions that happen in chemistry, and chemistry at an atomic level. So the Schrodinger equation, we have the Schrodinger equation from the quantum mechanics, and if you solve the many body Schrodinger problem using computers, then you have great scope to explain a lot of phenomena in chemistry used at atomic level. So that is an area people branched into, or theoretical physics actually started explaining phenomena in chemistry. And then similarly you have in biology, just like what theoretical physics did to chemistry, you have a similar phenomena where you have classical mechanics, you have classical force fields and Newton's equations, and that is explaining at a molecular level what is happening in various biological processes. So you have computational chemistry and computational biology, which is actually driven fundamentally by obviously the digital revolution, where you have you have computers and enormous computational power to solve quantum equations and classical Newtonian equations to explain chemistry and biology at atomic and molecular levels. So this is what we are going to see. So this is something we are going to start with computational chemistry. So what are we trying to do here? As I had said, we are trying to solve the many body Schrodinger problem. So when computers came about, people saw the opportunity to use computational power of computers to solve the many body Schrodinger problem, to explain chemistry at an atomic level. So few of the methods that came up were the Hartree-Fock method, and we have a series of methods that came up called the post-Hartree-Fock method, and then we had the density functional theory methods. So the Hartree-Fock method, had, so before that we must understand why we need numerical solutions to the many body Schrodinger equation. Now we need numerical solutions because we are trying to solve the wave function for multiple electrons. So when we take a system of atoms, or if we take a large compound, you have many electrons in that system, and it is not possible to solve analytically for that system. Thus it is not possible to solve the Schrodinger equation analytically for the system. So we are looking at numerical solutions. So the Schrodinger equation is a differential equation, and numerical solutions to Schrodinger equations within the finite range, we are going to have numerical integrators or differential equation solvers to solve this problem. Now in the first series of methods that came about, that is called the Hartree-Fock method. Now normally you would see that in a numerical integration, or when you are trying to solve a differential equation using numerical methods, you will perform an initial guess, and then you will iterate towards convergence, right? So here in our Schrodinger equation, the initial guess, you perform the initial guess for the potential of the system by considering the electrons as a Fermi gas. So if you consider the electron distribution about the nucleus as a Fermi gas, you can guess the close to the exact result. We can guess approximately what the potential would be, and then you put it into the equations, and the computer will iterate towards convergence. But the problem here is, with the Hartree-Fock, you have the problem of Pauli exclusion principle tells us that you have to consider also the spin of the electrons. So the spin, because of the spin of the electrons, if two electrons of the same spin are present close together, then they have a repulsion, and if they are if opposite spin electrons are present close together, then they have a attraction. Now this consideration must come into the picture, and Hartree-Fock method actually overestimates the energy because it does not handle the Pauli repulsion very well. So then came a series of post Hartree-Fock methods, and then we came to the density functional theory methods. Now in the density functional theory methods, you are not considering the wave function, but instead what you are trying to solve is the electron density distribution about the nucleus. And how we are trying to find the electron density distribution is, we are minimizing the Schrodinger equation, we are minimizing the distribution such that the energy is a minimum. And how we handle this electron correlation? So it's called the electron correlation, where we are talking about this Pauli repulsion or this attraction term. How we are handling this electron correlation is, we are introducing a separate term called the exchange correlation density functional, where we are trying to fit based on some experimental data, we are trying to fix that term. So just like I said, we consider the electron cloud distribution about the nucleus as an electron gas, we consider it as a Fermi gas, and we have an initial guess of the potential, and for that potential, we have a distribution of the electron gas or the Fermi gas, and we have a general form where we consider a homogeneous distribution and also a inhomogeneous distribution, and we have some undetermined parameters, and these undetermined parameters are figured out by fitting these functionals on some experimental data. So that's how the density functional theory improves the contribution, improves the accuracy of estimating the contribution of the exchange correlation term to the total energy. So with these methods, what can you find out? So this is the important thing, right? So with these methods, you can solve the electronic structure, determine the spectroscopic properties and other molecular and structural properties, and the thermodynamic properties. So what we are trying to do here is we are obtaining the structure for that corresponds to the optimized energy. So we are obtaining the right energy for that system, and when we have the right energy, we have the structure corresponding to the optimized energy, and based on those based on that data, we can extract the other properties that are of interest. So based on these properties that we are able to find out of the system, this leads to many applications such as new material discovery, and chemists also use it to understand reaction energy profiles and so on and so forth. So next we come to computational biology. So here we are dealing with large molecules where we are seeing proteins, DNAs and things like that, where it is not possible to apply quantum mechanics to solve these systems, so we turn to classical mechanics. So here we are trying to solve the Newton's equations. So in the Newton's equations we are having some. So when you're trying to solve the Newton's equations problem, you have to give the force on each term. So when you give the force on each on each particle, you can solve the trajectory for that particle and thereby track the dynamics. So we are trying to apply this problem to solve our protein interaction problem, or protein drug interaction, or protein ligand interaction it is called, or the protein DNA interaction problem. Now here, how are we going to do this? So we are going to have something called force fields. We are going to have something for force fields which tells us what is the force on this protein atom. In a protein we have large group of amino acids. Proteins are made up of amino acids, and in each amino acid there are many atoms, right? So on each atom we are going to tell what is the force. Based on that, the trajectory is computed for the entire protein, and based on that we can study the dynamics of the system and thereby understand the protein interaction or protein ligand interaction. The protein drug interaction is called the protein ligand interaction or the protein compound interaction, and then we have the protein DNA interaction. Now again, just like we saw in the previous case, here we have the force fields, but these force fields we are going to have some undetermined parameters that are determined by fitting the force fields to some experimental data. So in a force field you have many terms. You have terms that are contributions from the Coulombic interactions, the van der Waals interactions, and the bonded interactions, which are basically bonds between atoms which are approximated by the Hooke's law potentials. So you have all these terms, and the force on any atom is determined. So on these amino acids, they are determined by fitting this general force field to some experimental data, and we have some undetermined parameters in the force fields, and they are obtained by fitting these general force fields to the experimental data. Now obviously we are trying to use computers to solve these problems, the computational power of computers to numerically solve these the Newton's equations, and therefore this is computationally very expensive. So sometimes when the computational cost is too much, people turn to what is called molecular docking to study protein interaction and protein DNA interaction or protein ligand interaction. So here we similarly have some force fields or some or what are called scoring functions, and here we are interested to only estimate the free energy change. So we are trying to estimate the free energy change, and this is done based on some force fields or scoring functions which have some general form, and they are again there are some empirically fitted parameters that are obtained by, as I said, fitting that then the general equation to some experimental data. But as I said, this has limitation of only estimating the free energy change or the thermodynamic feasibility that drives these processes, but here we are computing the entire trajectory. So here we have more data for analysis or of the entire system's dynamics. So supposing I am doing a simulation of how a drug goes and binds to a protein. So I can see in the simulation, during the length of the simulation, how the drug goes and binds to the protein, and whether how long it stays in the active sites or whether it moves away from the active site of the protein and things like that. Now another area of computational biology is something called network modeling. Now in a biological system, we have the entire biological system is dependent on so many molecular level processes, and just like you would have studied in your, if you had taken biology in your 10th, 11th, 12th, you would have studied about the basic Krebs cycle. Now you know that is necessary for energy production, right? So now in that Krebs cycle you have a series of molecular level processes that happens, and supposing one process, so if one process in that network does not happen, now that affects many other networks, that affects many other molecular level processes that are necessary for the system to, so that affects many other biological processes, molecular level processes, for the system to function well. And in network modeling we study these network of pathways. So we have, so mainly in all these processes proteins are involved. Now there are certain enzymes that are necessary to activate these proteins, and supposing that function is not performed, now how that affects that pathway, and because of that pathway how the other pathways are getting affected. So that is something we do in network modeling, and the larger picture of this is called system biology. And so in system biology you have so many cellular you have so many molecular level processes that happen in a particular pathway, and the entire system evolves according to the these many cellular and the molecular level processes that happen in these networks and these pathways. So that is something we try to do. So if you are in this field, this is another word that you might hear many times. It's called the Monte Carlo simulation. So what is this? This is nothing but a numerical integration method. And here we let's say we'll take an example and then go to how it is actually used in the context of computational chemistry and biology. So here I am trying to find the area of this circle, and what if I'm trying to do this using Monte Carlo method, what I'm trying to do is I'm trying to take, let's say I'm trying to take 1,000 marbles and scatter them in this area. So this is the larger area that I have, that is the square, and I'm trying to calculate the area of this circle. Now I'll take 1,000 marbles and scatter them in the area of in the square, and I'll find how many marbles are placed within this circle, right? How many marbles are placed within this circle. Now since I'm taking 1,000 marbles and scattering them, and then these 1,000 marbles, now since the area of this area of this circle is now proportional to the number of marbles that are getting placed within this circle, right? Because they all, when I take 100 marbles and scatter them randomly, they all have equal probability of going and sitting anywhere in this whole area of the square, right? Now if the area of this region that I am interested to find occupies a larger measure of the area of the total area, then we will find that more marbles are sitting inside the that area. So therefore I can estimate this area of this circle in this way. So this is actually the basis of this Monte Carlo method. Now what we are trying to do is, here in a Monte Carlo method, when we are trying to apply it to computational chemistry or in computational biology context, we are now let's say I am trying to solve the Schrodinger equation, and as always in a numerical integration problem I have defined bounds, right? So here we had some defined bounds. Now within that defined bounds I am going to use what is called a random number generator, or it's called a making random walks. Now the random number generator is analogous to the analogy I gave of throwing 100 or 1000 marbles. Now when you have a random number generator, what it does is, for the Schrodinger equation, you make a small step, you take a small step and you see where, for that new step, in that new state, whether the energy is higher or lower than the previous state. Now if it is lower than the previous state, it will adopt the new state. If it is higher than the previous state, it will reject the new state and go back to the previous state. Now in this way, so this method is called the Metropolis sampling. Now just like how we sampled here, finding the marbles within this region, now this is how you determine whether for the random walks or the random number generator that we use, whether the states assumed are right or wrong. So this is the Metropolis sampling, and this is applied many times. So this is applied many times to solve the entire system. So the same approach is also applied to solve statistical mechanics problems, in the case of statistical ensembles, to obtain the partition function. So next we are going to move to the area of bioinformatics. So before that I want to give a brief, so bioinformatics basically deals with biological data and what insight we can gain from it. So we are basically talking about genomics data and proteomics data. So before that, little bit of biology background is necessary, right? So here now the very reason that we were able to apply classical mechanics and computational approaches to solve problems in biology is because of molecular biology, because now we are able to understand biological processes at a molecular level, and it is found that many molecular functions, all the molecular functions are performed by proteins. Now different proteins have different functions, and these different proteins are produced in the body by the genetic information that is present in our cells and in our largely in every living organism. Now this genetic information has the information of what proteins to produce, and when these proteins are produced, they perform different functions in different molecular pathways. Now this genetic information, as we know, is present in the DNA, and now the DNA has some sequence, right? So it has some nucleotide sequence. So you have U, so the 4 bases U, C, A and G. Okay, so when you have these 4 right, sorry, so here we actually have this is the base pairs of the RNA. So in the DNA, instead of the uracil we have the thymine. Okay, so instead of U we have T. So what happens is this DNA gets converted to RNA, and these RNAs get converted to proteins. Now, how do we know what protein to produce for what genetic information, or what DNA, or what? So how do you know that? How does the body know what proteins are to be synthesized? So it is based on these base pairs. Okay. So if so, three base pairs encode a particular uh, protein. So if you have U, it encodes this amino acid. Okay. So proteins are made up of amino acids. There are about 20 amino acids, and each amino acid is encoded by a set of nucleotides which are called codons. So three of them together are called codons, and these encode these proteins. Now, so this DNA first gets converted to RNA, and this RNA based on these codons are getting converted to are encoding some proteins, right? So this is the process. Now, not always that there are regions, there are what is called so there are parts of the DNA that never get converted to proteins. So we have the coding and the non-encoding regions, and sometimes genetic and environmental factors affect which part of this what part of the DNA or the genome is supposed to be converted to proteins. Okay. So we have enzymes that are involved in transcribing this genetic information into proteins, and those are called transcriptomes. Okay. So as I said, there are several enzymes involved in transcribing what this DNA into proteins, and these enzymes that help in this transcribing are called transcriptional factors. They control which part should be expressed and which is not, and this data of this data of which part of the DNA or the genetic material is expressed into RNA is called the transcriptomics, and then the proteins that are obtained from it is called the proteomics. Okay. So we have genomics, transcriptomics, and proteomics. So what insight can we gain from this data? So that is the main interest in bioinformatics. So supposing I have some genomic data, so what insights can I gain from it? So people are trying to understand the phylogenetic relationships. That is, if I have some genomic data, I compare it with the genomic data of other organisms and see how they are evolutionarily related. So similarity in some kind of sense, the evolutionary family and things like that. And another area of application is so supposing I have the genomic data of, let's say I have sequenced my genomic data and I'm comparing it with somebody who has some disease. Okay. So what is the disease risk that my genomic data possesses, right? So in diagnosis, so in predicting disease risk, people are using these kind of approaches, and this is where all the new data science and machine learning and the AI approaches are coming in to use this data to make these predictions. So in disease risk prediction and in prognosis, in the sense that supposing I'm being treated for a particular disease, now based on the treatment I've received, if I've improved or if I have regressed, or what is the prognosis prediction, right? So that is also based on some data. And in precision and personalized medicine, so in cases where now not all of us so supposing I'm taking, so let's say 500 mg of paracetamol, now all of us are going to have little different pharmacokinetics, pharmacodynamics of that drug in our body, because our genetic information is different. So that's why you have the space of this personalized and precision medicine. So I left out a point here. So also in terms of the pathway analysis, so I have some, let's say I have some disease, and I'm comparing my pathways and my pathways to somebody who does not have the disease. So how are the pathways altered? So how does the gene expression vary in somebody who has the disease and somebody who does not have the disease? So these kind of analysis gives some useful insights, some actionable insights. So when we come to proteomics, we are talking about protein data, so amino acid sequences and things like that. So here again we are trying to understand the protein structure and function based on some similarity. So supposing for some protein, I know the structure, I know the function. Now I am taking a unknown for a protein for which I don't know the structure and the function, I just have the amino acid sequence. So if it is similar to some protein for which I know the structure and function, if the amino acid sequences are similar, then I can make a guess or a prediction that it is going to have some kind of a similar function and a structure. So this is obviously has a lot of applications in the area of drug discovery or enzyme engineering, where people are trying to make enzymes for different processes, like say a textile that involves textile or brewery industry or things like that. So similarly in chem informatics, we are trying to understand, or we are trying to gain insights into chemical data. So we have databases of chemical compounds. So many structural molecular properties of these compounds are available, and also their sometimes the experimental biological activities or the material properties are available. So what we are trying to do is, most of the time we are trying to understand the relationship between the structure and molecular properties and the property of the material. So the physical property of the material and the structure and the molecular level properties, and this leads many times to new material discovery. And so we are trying to understand some kind of statistical way how these molecular and structural properties are related to the physical property of interest. Let's say if we are doing some solar cell research, or we are trying to understand how the efficiency of the solar cell material is related to some kind of molecular level properties of that material, and we are having some we are trying to gain some statistical correlation through this from this data, and we are trying to enumerate based on this statistical relationship. We are trying to enumerate on compounds that will possess this desired property, and that leads to new material discovery. So similarly in the biology realm, we are trying to identify new small drug molecules. We are trying to understand how the molecular level properties of each compound play an important role in determining the biological activity. So we try to understand through a statistical way, establish some statistical correlation between the structural and the molecular properties of those compounds and the biological activity, and then again enumerate on compounds possessing those desired biological activity, and this leads to new drug discovery. So in these spaces of bioinformatics and chem informatics, which is largely data driven, now we see the application of data science, machine learning and AI approaches which are data driven. So we're going to see